The last thing we're going to be discussing in today's lecture is a technique called polymerase chain reaction, or PCR. PCR is essentially the DNA replication reaction that we're doing in the laboratory. We refer to this as DNA replication that is done in vitro. Now, if you're not familiar with this phrase, in vitro, this translates as meaning in glass. And the reason we say it is because we're describing something that is happening not inside of a living cell, but outside of the cell, in the laboratory. So anytime you hear something described as being in vitro, you should understand that that thing that's happening is happening outside of the living cell. PCR is DNA replication occurring in vitro. What happens in PCR is we have a piece of DNA, a region of DNA that we're interested in. It's usually a gene. And we use PCR to replicate that gene over and over and over and over again. We describe this as amplifying the DNA. And the reason you would want to amplify a gene, make many, many copies of it, is because when we have many copies of a gene, we can do some further investigation of that gene in the laboratory. And one of the things we want to do very frequently is figure out the order of the bases in that gene. When we're first learning about a gene, one of the very first things that we need to do is determine that sequence. Once we know the sequence of the bases in a gene, we can take that to our genetic code table and we can figure out what protein is being made from that gene. Sometimes when we're performing PCR and amplifying a gene, we're trying to determine if that gene is present. In other words, you may discover a gene in one type of organism, and you may be curious whether or not that gene exists in another type of organism. So you can use PCR to probe for that gene. If it's present, you'll amplify it. You'll make many, many copies of it. If it's not present, you won't. PCR is a technique that can create millions of copies of a gene or other section of DNA. Now, PCR requires a very particular enzyme, a special form of the DNA polymerase enzyme. And this special form is referred to as Taq polymerase, TAQ. PCR also requires a machine, and that machine is called a thermocycler. This is an image of a PCR machine. It's about as big as a large toaster oven or a small microwave. PCR is such a common technique now in so many different types of labs, both research labs and clinical laboratories, that you see thermocyclers all over the place. Thermocyclers are not inexpensive, but they have certainly come down in price as PCR has gotten more and more commonplace. A typical thermocycler in 2020 would run you somewhere between $3,000 and $6,000. The PCR technique takes advantage of the natural process of DNA replication because of the complementary nature of DNA. Remember, inside the cell, the DNA polymerase enzyme is able to make new DNA because it has some old DNA to use as a template. In PCR, the same thing is happening. Some old DNA is being put into the reaction. The Taq polymerase enzyme can use that old DNA to build new DNA. So let's look at the steps in the PCR process and the temperature changes that are occurring at each step. Step 1 in PCR is called the denaturation step. In this first part of the reaction, we're going to heat the thermocycler up to 94 degrees centigrade. That's a very high temperature. At that temperature, the DNA that we put into the reaction tube is going to separate into two strands. It's going to denature. Remember, inside a living cell, an enzyme would do this. But during PCR, we're going to use heat to do this because heat is capable of denaturing DNA. In the next step, we're going to lower the temperature down to about 54 degrees centigrade. Now that's still pretty hot, but that lower temperature is going to allow primers to attach to the individual strands of DNA in the reaction tube. Remember, inside a living cell, an enzyme is going to build RNA primers to put onto the template strands of DNA. In the PCR reaction, we're going to add primers pre-made, and those primers are going to be made out of DNA instead of RNA. We're going to add pre-made primers, and we're going to add them in pairs. One primer is going to anneal or attach to each of the two strands of original DNA. In step 3 of the PCR reaction, we're going to again heat up the reaction tube to about 72 degrees centigrade. This is the extension step. This is the optimal temperature for our Taq polymerase enzyme to build DNA. After enough time has been allowed for the DNA to be completely replicated, we're going to repeat the entire process. We're going to heat the tube back up to 94 degrees centigrade, and any double-stranded DNA that is inside that reaction tube is going to denature. Then we're going to lower the temperature to allow primer annealing. Then we're going to raise the temperature to allow extension. Then we're going to repeat it. Separate all the double-stranded molecules of DNA, allow primer annealing, allow extension. And it just goes on and on, over and over again. Each combination of steps 1, 2, and 3 is referred to as a cycle of PCR. And we typically run 30 to 40 cycles inside of a thermocycler for each PCR reaction. That takes anywhere from 3 to 5 hours of time. Now, I want to address why we need this special form of DNA polymerase for PCR. Remember, the first step involves us heating up the reaction tube to 94 degrees centigrade. That's the temperature that's required to denature DNA, to separate the two strands of DNA. The problem is, at that temperature, you will also denature any proteins that are in the tube. Remember, we said when you denature a protein, you unfold it, and that's going to make the protein unable to perform its job. So if we were to put ordinary DNA polymerase into a PCR reaction, it would get denatured during step 1. Instead, we're going to use a form of DNA polymerase that evolved inside an organism that lives at temperatures in the area of 94 degrees centigrade. This particular organism is called Thermus aquaticus. So we took the T from thermus and the AQ from aquaticus, and we came up with Taq polymerase. Thermus aquaticus lives in thermal vents on the floor of the deepest parts of the ocean. Thermus aquaticus lives at that high temperature, and so all of its enzymes are adapted to function optimally at that high temperature. You probably could guess that Thermus aquaticus is an archaea. So PCR is going to replicate DNA, whatever particular area of a DNA molecule we're interested in amplifying. And again, it's typically a gene, but not always. The nature of the PCR reaction is exponential, and that's what allows us to make millions of copies of a piece of DNA over a relatively short amount of time. Inside that test tube, with every cycle, we're going to double the number of DNA molecules. So we begin, let's say, with one piece of DNA, double-stranded. We're going to send it through the cycle. We're going to separate the strands, that's denaturation. Then we're going to put the primers on, that's primer annealing. Then we're going to build new DNA, that's extension. Now we've gone from one double-stranded piece of DNA to two double-stranded pieces of DNA. The process repeats. We're going to go from 2 to 4. Then the process repeats. We go from 4 to 8, and then to 16, and 32, and 64. It's an exponential process. And that's what you can see down here on the bottom of this slide. In very short order, we get millions of copies of that piece of DNA that we're interested in. It is an exponential amplification. PCR has really revolutionized the work that gets done in laboratories. It's one of those advancements that comes along every so often that really pushes science forward by a tremendous leap. It turns out that the test that we are using to diagnose SARS-CoV-2 infection, remember that's the virus that causes COVID-19, that diagnostic test is a PCR test. Now, if you recall, we said that this virus is actually an RNA virus. It doesn't have DNA inside of it. So we have to put an extra step into the PCR process. And we can do that because of our understanding of some other RNA viruses, including the HIV virus. Remember, there's an enzyme called reverse transcriptase. HIV uses that enzyme to help it hijack human cells for replication purposes. The reverse transcriptase enzyme, like the name suggests, is able to do transcription in reverse. Remember, the process where DNA is used to make RNA is called transcription. So reverse transcriptase takes RNA and uses it to make DNA. So what that's going to do is take any RNA that might be present in the patient sample and use it to build DNA. And then, in order to determine whether or not the correct DNA is present, the SARS-CoV-2 DNA, we probe for that particular sequence of bases with primers that were designed for that virus. There are actually 3 different primer mixes, or what we call primer probe mixes. I've got them listed here, but don't worry about this information. This is very technical information. What scientists had to do was take that RNA out of SARS-CoV-2, figure out the order of the bases in that RNA, and then figure out what a DNA would look like if it was made from that RNA. Remember the complementary nature of base pairing. And then once scientists knew what kind of DNA could be made, then they could look for particular areas in that DNA that acted as genes and could code for proteins. And they made the primers to find and anneal to those genes. There's a protein that's referred to as the nucleocapsid. Remember, that's the outer coating, the capsid, the outer protein coating around a viral particle. And they've targeted that gene with a primer. There's also, interestingly enough, a primer in there that acts as a control. What it does is it detects any human nucleic acids. In other words, it makes sure that if we get a positive test, it's because of viral DNA and not human DNA. The RT, reverse transcriptase, PCR test that we're using for this new coronavirus is a good test, but it's not a perfect test. There have been some problems that you may have heard of in news stories. You have to remember that a test, a diagnostic test, is only as good as the materials that are used for that test. Early on, there were some issues with the primers. The test had to be reworked because the primers were not correct. We also have had issues with the samples that are being collected from people. Remember, in order to collect this virus, you have to get a swab way back into the rear area of a person's nasal cavity, and that's not easy. So if an incorrect swabbing is performed, you're not going to see any viral particles. It doesn't mean that the patient doesn't have the virus. It just means that you didn't collect any viral particles on the swab. So it's not a perfect test, but it is the best test we have right now to diagnose someone with this very dangerous virus.